Hi, welcome to the WellnessFX baseline panel video where we specifically talk about glucose and hemoglobin A1C. Now, in order to talk about those, let's just really review what glucose even is in the first place, including the different pathways that it can be utilized in the body and what really its role is in terms of getting into a cell, producing energy, and the mechanisms involved in getting glucose into the cells. So glucose is really just a 6-carbon monosaccharide or monomer, which basically means it's a real simple sugar. It's just a single sugar molecule. You may have heard of things like sucrose, which is considered to be a disaccharide, and all that really is just 2 monosaccharides linked together. Now, you may also have heard of things like polysaccharides, meaning many sugars, and things like cellulose, starch, and glycogen are all examples of polysaccharides. Now, what a polysaccharide really is, like if this were glycogen, is essentially to take a glucose molecule and just combine it with a bunch of other glucose molecules into this much larger, more complex structure. Now, we consume, generally, polysaccharides, unless you're consuming a lot of glucose or perhaps sucrose. Many things that we'll be consuming, if it were a potato, for example, would be more in a starch form or in a polysaccharide form. Now, these polysaccharides are broken down a little bit in the mouth via salivary amylase, starts to break off some of those 1,4-glycosidic bonds that holds these glucose molecules together, makes its way down past the stomach, eventually into the small intestine, where it's further broken down into what is essentially single sugar units or monosaccharides to be absorbed past your gastrointestinal tract and ultimately into you. Now, the first place that it goes, which is important to discuss, is from the small intestine. You absorb glucose, which is the simple sugar we'll be talking about here, into the liver. Now, this is good because what you'll find is excess levels of glucose in the bloodstream is very problematic. So the body wants to try to minimize the amount of glucose that actually goes to your body, into your bloodstream, so it first sends it to the liver. Now, your liver, with these monosaccharides or glucose molecules, can link them together and create its own polysaccharide known as glycogen. And glycogen is just simply a storage form of glucose in the body. Your body can use glucose molecules. Right now, it can't use this. This is too large and too complex a molecule to utilize, so it's considered to be the storage form of glucose in your body. And under the right circumstances, the body can produce hormones that can increase certain enzymes that will snip all these bonds, basically, again, making glucose available to your body if it actually needs it. So the first place it goes to is the liver. Now, the liver, hopefully, if it's not too much glucose, will store much of it in the form of glycogen. But the remaining glucose actually goes out into your system and into the bloodstream. Now, here what we have is glucose in a blood vessel, and here we have a cell. Now, what we want to try to do is get glucose from the bloodstream and into the cell. Now, in terms of quantities of cells, this type of cell is really representing skeletal muscle or adipocytes or fat cells as well. It's really the same mechanism for both these types, and because they're so abundant, we'll just use those as a model. Glucose is now in the bloodstream. You've consumed a meal, the liver passed off some of it through into the blood. Now glucose is in the bloodstream. We want to get it inside of the cell so that we can make ATP, or energy. We won't get into the biochemical pathways of that. What we're more interested in is how glucose gets from the bloodstream into the cells of our body. Now, we have an organ called the pancreas. The pancreas, in the presence of glucose in the blood, produces a hormone called insulin. Now, glucose cannot get past this cell membrane by itself. It needs help. Fortunately, on the inside of this cell, you have these little vesicles, or what you could just imagine as tubes. These are glucose transporting tubes, and we somehow need to get these glucose transporting tubes, specifically in adipose tissue and skeletal muscle cells, called GLUT4 or GLUT4 vesicles, or glucose transporters, from the inside of the cell to the cell membrane, so now glucose can come in. Right now it can't. And here's essentially how it works, and there's a very complex process that occurs inside of the cell. In the presence of glucose in the bloodstream, the pancreas senses this and produces the hormone insulin. Now, insulin itself does also not get into the cell, but rather binds onto what's called an insulin receptor, abbreviated IR here. So insulin binds onto this insulin receptor here, which then creates a series of different events intracellularly or inside of the cell, which we won't get into here, but that essentially have the effect of what's called translocating or moving these glucose transporters, these tubes, into the cell membrane so that now glucose can come in. So under the influence of insulin, when insulin binds onto the insulin receptor, a number of series of events take place inside of the cell that ultimately has this vesicle, GLUT4 vesicle, move these glucose transporting tubes to the cell membrane so that now glucose can enter into the cell. When glucose enters into the cell, again, biochemically, glucose can be split through glycolysis and making pyruvate, eventually getting into the mitochondria to make ATP. Glucose is a fantastic way, if working properly, to make ATP so that all the cells of our body can do what they do and we ultimately can run healthy and properly. Now, here's a couple other things in terms of just big picture of what glucose, the different pathways that glucose can be involved in. One is, as we've just identified, if glucose can get into a cell, it can make ATP. Glucose, if it's not currently needed, as we've talked about in the liver, skeletal muscle cells and the heart can do this as well, but if you have excess glucose and an organ or like the heart or the skeletal muscle, if it doesn't have too much glycogen, then glucose can be converted into this polysaccharide called glycogen and stored within an organ like the heart for future use if glucose ever drops too low. Conversely, you notice there's an arrow going back. Glycogen can also get broken down to produce more glucose if ever necessary, if there's a period of time where we have periods of low glucose. And I'll give you a real classic example is overnight. When you eat dinner and you go to bed, you may have a period of time, anywhere between 12 and 14 hours, where you're not consuming any more glucose. Your body will have utilized all the glucose from that meal many hours ago, but we still need it, which we'll talk about in a bit. Your body can actually break down stored glucose as this polysaccharide to produce more in order to help fuel the body and keep the cells making ATP that really need it. Now, in extreme cases, if we don't have much glycogen available, your body can actually take a non-glucose compound, something like lactate or pyruvate, some amino acids, for example. Something that wasn't glucose originally can actually be turned into glucose via a process called gluconeogenesis. You're making new glucose from something that wasn't previously glucose. The primary hormone that's involved with getting glucose from the bloodstream into the cells is insulin. On the other hand, there are periods of time when glucose can actually be too low. The number one organ that is most impacted by periods of low glucose is the brain, the nervous system. As I mentioned before, the heart actually stores glucose, or stores glycogen, which is good, because if there's ever a period where your glucose starts to drop, you don't want your heart to lack the fuel to keep pumping. So it stores glycogen so that if your glucose goes down, can liberate the glucose molecules from glycogen in order to help keep the cardiac muscle cells continuing to beat, continuing to utilize ATP, and to have your heart keep pumping. The brain does not store glycogen, however. And if you've ever experienced yourself or have anybody, you know somebody that experiences this, periods of low blood sugar cause symptoms like agitation or irritability, sometimes brain fog or an inability to concentrate. In some people it might be fatigue, for example, as well. Many of those things, irritability, agitation, as well as an inability to concentrate, that's all brain-based. So more than any other organ in your body, you need to maintain healthy, stable levels of glucose. To do so, there are 4 primary hormones that can actually raise glucose if your glucose ever drops too low. Those are glucagon, also made by the pancreas. And as the name glucagon would suggest, glucagon, glucose is gone, so that the pancreas makes glucagon so that you can liberate some of your stored glucose or make new glucose to help keep glucose stable, specifically for brain function. Another one is cortisol, made from the adrenal glands. That cortisol is also known as a glucocorticoid. And again, as its name would insinuate, cortisol is used in glucose regulation, specifically to bring glucose levels up if they ever drop too low. Another one is growth hormone, which we really won't talk too much about, And then the last one are things like the catecholamines made by the adrenal medulla, specifically things like epinephrine and norepinephrine. Now, if anybody ever has periods of low blood sugar and they start to get shaky, for example, between meals, that's a pretty strong indication that their blood sugar started to drop. Their body saw that as an emergency, specifically the brain saw that as an emergency, told the adrenal glands, the adrenal medulla, to make epinephrine in order to raise glucose in order to help really save the brain from damage. So those are 4 of the hormones that are involved with raising glucose if glucose ever gets low, and this is insulin. If glucose is high, will help regulate and try to get it inside the cells. Now in the next part of the video, we'll talk about what happens when glucose is too high or too low, what that means, as well as a really great marker for looking at blood sugar regulation called hemoglobin A1C. So now that we talked a little bit about the physiological roles of glucose, some of the different pathways that are involved, let's talk about really now a blood chemistry test. Now glucose, I will start by saying, is a transient marker, meaning it's not terribly stable marker. Now it deserves to be on every single blood chemistry because it can definitely tell you what's going on with blood sugar regulation, but there are some issues with it as well. And I'll just simply state this. Now, and this is not something that you'll need to ask or talk with every single client. But understand that glucose is the preferred fuel for the cells of your body to make ATP. Therefore, it's very much involved with a stress response. For example, if somebody has a stress response, things like epinephrine and cortisol tend to be elevated, and both of those will tend to elevate glucose, which, to be honest, during a stress response you actually want. You want all the glucose or fuel available to really have what's called a fight or flight response in order to get yourself out of a bad situation. So high glucose during a stress response is desirable. But let's say, for example, if somebody was in fact fasting, which, as we talked about before, is critical for getting an accurate glucose measurement. Let's say, for example, somebody was going to get their blood drawn for the WellnessFX panel and that morning almost got into a car accident, or maybe got pulled over, or had a horrible argument with their spouse as they were leaving. Or even, and that would have been before the lab visit. Or at the lab itself, maybe somebody just hates needles, almost passes out when they get their blood drawn. Or perhaps the phlebotomist missed the vein a few times and had to use the other arm. In any of those cases, that could be sufficient enough to create a stress response in the person. Meaning that if somebody on their blood chemistry has a glucose level of 95, while that may be within the normal reference range, what you don't know is, did somebody, if they almost got into an accident on the way to the lab, had their glucose go way up, and you're seeing 95, which is actually back, the body's regulating it and it's bringing it back down, or maybe in the lab itself, maybe somebody has a lower glucose and this is on its way up. Point is, you can't look at a blood chemistry without having glucose on it because it's such an important marker. But it can move around quite a bit. And understand that if you see some results on glucose that are a little bit confusing, that it may be that there was other factors involved with it, which is why it's great to be able to look at other markers on the blood chemistry test to really see what's going on in terms of somebody's glucose. So, glucose levels, if elevated, generally means that for some reason there's too much glucose in the bloodstream, and for whatever reason it's not getting out into the cells. Now, reasons for this. Obviously, if somebody wasn't fasting, it could be because they ingested it, but that's not the case with blood chemistries because people should be fasting. Another possibility is that their body's actually making too much of it, either through the liberation of glycogen or gluconeogenesis like we talked about, that maybe their body is making too much of it. But if you see it high on a blood chemistry test, then it basically insinuates that for some reason glucose there's too much of it in the blood for some reason. Now, looking at this a little bit more specifically, then the question is, and in some of the more advanced WellnessFX panels we can look into this more deeply, but the question is why? Why is the glucose in such high amounts in the blood? It's not getting into the cells, but why is it not getting into the cells? It may be that in some people, the pancreas is not making sufficient insulin to stimulate this response to get glucose into the cells. That's one possibility. Another possibility is the pancreas is making insulin, but there's some defect at the receptor site, which is usually referred to as insulin resistance, or something else that's happening post-receptor within the cell itself. In either case, glucose will be unable to get in. If you're not making insulin, glucose won't be, this process doesn't take place, glucose can't come in. Or if insulin is being made, if there's a cellular issue either with the receptor or afterwards, again, glucose won't come in. It's really essentially the same thing, that glucose is in the bloodstream too much for some reason, but it's for pretty different purposes. One other quick note I meant to mention about the GLUT4 vesicles. This is an insulin-dependent process, meaning you need insulin in order to get this GLUT4 vesicle to translocate to the membrane to bring glucose in. Something that is critically important to understand, however, is in skeletal muscles, these GLUT4 vesicles also can be translocated to the membrane via an insulin-independent process, meaning there's no insulin required to do so. And that is skeletal muscle contraction, otherwise known as exercise. Simply contracting your muscles against resistance, the skeletal muscle contraction, allows the GLUT4 vesicles to translocate, bring glucose in, and not require insulin. So this means that if there's any clients or patients that have elevated glucose or A1C, which we'll talk about, one of the things that they absolutely have to be doing is exercise. And if they tell you they are exercising, there may be some pretty significant physiological issues going on, but it may be that they're not exercising enough or with enough intensity to actually help facilitate this insulin-independent process of getting the GLUT4 vesicles to allow glucose to actually come in. It's a very key, important point. So if glucose is high, generally it's referred to as insulin resistance, and it's due to poor diet, sedentary lifestyle, or possibly some other physiological things that are going on in the body. If glucose is ever low, below the lab range, then what you're looking at is probably what could be referred to as either reactive hypoglycemia, or if it's low enough, actually diagnosable hypoglycemia by a licensed medical doctor. If glucose is low, the primary reason for it, and this is why I covered those 4 hormones that are involved with raising glucose, is some issue with one or more of those particular hormones. So if somebody's glucose starts to drop, the brain wants glucose to be stable. And if it's too low, one of the reasons why is because either glucagon, cortisol, growth hormone, or the catecholamines are not being secreted enough to help maintain glucose at a stable level. The other thing that you actually may be seeing in low glucose is what could be called blood sugar fluctuations. Now, yes, in the periods of low glucose, perhaps it's not that one of those 4 hormones are actually helping to keep glucose at a stable level, but maybe somebody has these excessive blood sugar fluctuations because, for example, they're producing too much insulin and it's doing too good of a job, if you will, to get glucose out of the bloodstream and into the cells. Now, this is actually thought to be very, very early insulin resistance, that over time, these periods of excess insulin actually start to break down the insulin receptor, have it not work as well, and eventually this person may end up with high glucose and insulin-resistant type tendencies. So if somebody has low blood sugar or low glucose on their lab, one of the things that you'd want to ask them to see if it was a spurious result or if it was tended to be true for that individual, is to ask them questions. If you skip a meal, how do you feel? Do you feel agitated, jittery, lightheaded, irritable, for example, between a meal when you're fasting? Very often, people that have reactive hypoglycemia are also not very hungry in the morning. People that have this tendencies towards low glucose, hypoglycemia, that they will often say that after they eat, they feel energized, and that's actually not the way we're supposed to feel. We should feel hungry, eat, and then feel not hungry any longer. We shouldn't feel tired, or we shouldn't feel energized after a meal. If somebody feels energized after a meal, then that may be this low glucose is in fact something that they suffer from more often than not. Now, I mentioned how glucose is not necessarily the most stable marker, and it can really move around quite rapidly. There's another marker called hemoglobin A1C that definitely needs to be talked about. Now, there's a process, it's just a chemical process called glycation or glycosylation. And all it really is when a sugar combines with a protein and forms what's called an advanced glycated end product, or AGE. This process, is just a chemical process called glycation or glycosylation. Now, this is happening in our body all the time because we have proteins in our blood, as we talked about, and you have sugar in the form of glucose. So this process is going to take place. The more sugar you have, the more glucose you have in the blood, the more this process is going to take place, and the more of these advanced glycated end products will be produced. Red blood cells have a lifespan of about 120 days, or about 3 months. Now, you've heard of hemoglobin. Hemoglobin is the oxygen-binding component inside of a red blood cell. Hemoglobin is really made up of heme, which is iron and an organic molecule, and globin, which is a protein chain. So it's made out in part of protein. The more glucose that somebody has circulating in their blood, the more this glycation process can take place, and the more that if this protein is the protein found on hemoglobin, then what you get is this marker called hemoglobin A1C, also known as glycosylated or glycated hemoglobin. In other words, that means hemoglobin that has gone through this glycation process with sugar that's found in the blood, or glucose that's found in the blood. Now, you could technically look at other proteins found in the blood to find out their level of glycation. But it's the lifespan of the red blood cells that makes it such a valuable marker. For example, let's say you had a protein that only the lifespan was maybe 2 or 3 weeks. If you look at the amount of glycation that happens with that particular protein, you're only looking at blood sugar management for really those 2 or 3 weeks. For example, if glucose was really high for that 2 or 3 week period, there'd be a lot of that protein that was glycated or glycosylated, but it's only for that short window. Hemoglobin A1C, because red blood cells have a lifespan of about 3 months or 120 days, because hemoglobin is found within red blood cells, and because part of hemoglobin is a protein, makes hemoglobin A1C a marker of 3 months worth of blood sugar management. And that's why this is such a valuable marker when compared to glucose by itself. Now, I can tell you on a personal level, I have seen individuals with an elevated hemoglobin A1C, but normal glucose, and sometimes even low. So that's why glucose is a good marker, but it's not a great marker. Hemoglobin A1C is a great marker at really evaluating blood sugar management for a long period of time, specifically about 2 to 3 months, more on the 3-month range. If hemoglobin A1C is high, then what you're really looking at is too much glucose in the bloodstream. It's therefore ability to glycate some proteins, the ones specifically found on hemoglobin, making hemoglobin A1C high, and one of these processes or both is taking place. Either somebody is not making insulin and therefore not getting glucose out of the bloodstream and into the cells, or they're making insulin and they have insulin resistance, in which case insulin's not working well and you're not getting the glucose or the GLUT4 vesicles, transporter vesicles, bringing glucose in. Another thing is, if you see hemoglobin A1C high, one assumption that you can make that's fairly accurate is that the person is not doing enough exercise or intense enough exercise to actually stimulate this insulin-independent process of getting GLUT4 vesicles to the membrane to allow glucose to come in. So, what do? If somebody has high glucose, you first jump down to the hemoglobin A1C to see if, too, it is high. If glucose is high but the A1C is normal, then it may be that glucose being slightly elevated was due to something else. This is a far more stable and reliable marker. Generally speaking, if this is normal but glucose is high, you may be seeing very early insulin resistance, but chances are that is not so much of an issue for this given individual. If the hemoglobin A1C is high, we pretty much know what we need to do for those particular individuals. They need to follow a better diet that is high fiber, relatively higher in protein, low in processed or refined sugars and carbohydrates. Try to eat glycemically balanced meals fairly regularly throughout the day, 3, maybe 4, or even 5 glycemically balanced meals, again, high fiber diet, as well as exercise that contracts the skeletal muscles. Walking, for example, is a great exercise, but I don't know if that's enough to actually stimulate this insulin-independent process to take place. They may need to do some more high-intensity type training or some resistance type training using weights or something like that. There's also a number of great supplements that can be used to help augment or correct some of these physiological imbalances that are going on. For example, there are some nutrients or herbs that actually block glucose absorption while you consume it, so you get less glucose actually coming into your system in the first place. There are some other nutrients or herbs that help stimulate the pancreas to make more insulin. There are some that help regenerate what are called the beta cells of the pancreas, and those are the ones responsible for making insulin. There are many that have actually been shown to either sensitize the receptors to insulin or help some of the intracellular processes to make this process take place, including allowing the GLUT4 transporters to actually go to the cell membrane. There's a number of different things. If somebody has low glucose, chances are, and again, you've asked them to make sure that they have irritability, agitation, maybe jitteriness, brain fog, fatigue in between meals, and are energized by meals. If that's their tendency, then you can be pretty sure that their glucose may be a little bit lower than it should be, that some of those 4 hormones are not doing their job adequately. And the best recommendation for these individuals is to eat as if it was an IV infusion of glucose, meaning they can't keep glucose stable themselves, so they need to do it exogenously via their meals. They need to eat every 2 to 3 hours, usually a somewhat small, glycemically balanced, for example, not crackers, not bread, similar in quantity, or I should say quality, to what we talked about with high glucose, but they need to be eating very, very frequently throughout the day in order to help maintain stable glucose levels orally, because they can't do it physiologically or hormonally themselves. Even deeper than that, it requires digging in and finding out why those hormones aren't working, but for our purposes here, just make sure they're eating every 2 to 3 hours. They have to eat first thing in the morning. They're very often not hungry, but these are people that should be eating something for breakfast, eat a glycemically balanced meal, and again, as often as every 2 to 3 hours, to make sure that they can stabilize their glucose levels themselves. So I hope you found this video helpful, and I look forward to seeing you in the next video.